So welcome everybody to this conference today. It is my absolute pleasure to have Nelly Fitopoulos and Christiana Salmon from the neuro, I'm going to say. I think Christiana is probably got a broader umbrella under her. They are both working in research related to ALS and genetics, and they'll explain a little bit better their roles as the presentation begins. So it's my absolute pleasure to begin this conference on genetics and ALS with Nelly and with Christiana. I let you take it away.
Thanks, Kate. And I'm going to let Nelly kick things off and start with her portion of the presentation.
Okay, well, hello everyone. It is a pleasure to be here today and to present to all of you. My name is Nelly Fitopoulos. I'm a genetic counselor at the Montreal Neurological Institute. Before I started working as a genetic counselor, I did research in the field of neuroimaging genetics. So that's focusing on the genes that are involved in brain structure. And I did that at the Douglas Institute and then went on to do the genetic counseling degree. So that's where I am now. I just wanted to also show that I work alongside Nancy and Noha. She's here today, and that's another genetic counselor and my mentor at this time at the Neuro. So the talk will consist mainly of two parts. The first part will be a little bit of a description of what genetic counseling is and what we do as genetic counselors. Talk a little bit about genetics. So I'm not sure the background for everyone in this talk, but just so that we're all on the same page. And then we'll talk a little bit about genetic testing and what the indications for genetic testing are, what to consider before going or agreeing to go forth with genetic testing, and what are the possible results that could come out of genetic testing. And then the talk will be transferred over to Christiana, who will talk a little bit more about the specific genetics in ALS and the therapeutic strategies that are currently available. And then we'll also have a question period at the end. So if you hear me out of breath, I apologize. I'm 8 months pregnant, so I get out of breath. And also the pregnancy brain is a thing. So if ever something is not clear in the presentation, please do not hesitate to interrupt me and ask me to repeat something. I apologize in advance if I repeat myself, but just please, this presentation is for you, so don't hesitate. So genetic counselors, who are we? So we're healthcare professionals. It is an advanced degree. It's a master's degree that specializes particularly in genetic counseling, and it combines the two fields of medical genetics and counseling. So it's a supportive, evidence-based approach. Most of us are certified by either the Canadian Association of Genetic Counselors or the American Board of Genetic Counseling. So just to give you an idea, it's a little bit similar, like if you go to law school and then you do the bar exam, this is, you do the program and then write this exam after, and it's a way of assuring that all of the genetic counselors have the minimal amount of knowledge to practice. So genetic counselors can help you in various different situations. This can be about deciding whether or not genetic testing or genetic screening is appropriate and beneficial for you and your family. Sometimes with people who have relatives who are living with a genetic condition, this can also impact their decision to either have children or the choices for family planning. Sometimes individuals who are related to people with a genetic condition also need support. So we can offer that to the caregivers and also estimate risk for relatives who are related to individuals with a genetic condition such as ALS. Sometimes it's not as relevant perhaps in neurology, but some genetic conditions, especially for children, are extremely rare. So sometimes we help connect different families to each other in order to have that kind of support, and then also to make connections with the community and supportive services. At this time, the working definition for genetic counseling brought forth by the National Society of Genetic Counselors is that genetic counseling is the process of helping people understand and adapt to the medical, psychological, and familial implications of genetic contributions to disease. So it's not so much focused on each disease itself, but what the genetic components of that disease can be. And we help individuals and their families incorporate that information into their lives. An important distinction, because now we see more and more that genetic testing is becoming available, especially directly to consumers, genetic testing. It is an important distinction between genetic counseling and genetic testing. Genetic testing is a tool that genetic counselors may or may not use in terms of providing a service, but the genetic counseling is a much larger process that can involve genetic testing, but they're not synonymous. So if you are interested in consulting a genetic counselor, to know that it is part of the healthcare system. It's not necessarily accessible equally across Canada. It does depend on province. And that's actually something that Christiana just recently published. I saw this was published yesterday, so she can definitely tell you more about this. But it is part of the healthcare system. And usually how this happens is through a referral of your treating physician or specialist. So this could be a neurologist, especially in ALS. But you can also self-refer if there's been a diagnosis and a gene identified in your family and you're a relative of this person, you can self-refer to our services as well. So you may want to see a genetic counselor, as I mentioned, if you're diagnosed or at risk for a genetic condition such as ALS. Genetic conditions, just to give you a broad idea as well, can extend far beyond neurology. It could be in cancer, pediatrics, cardiology, etc. If you would like to see a genetic counselor if you have a family history of a genetic condition, but you're not necessarily having symptoms yourself. Sometimes genetic counselors can do risk assessments for individuals that are consanguineous, so if they're related to their partner other than through marriage. Some couples experience multiple miscarriages. So unfortunately, miscarriages are common, but if they're recurrent and there's more than 3, this can be an indication to see a genetic counselor. And there are also certain ethnic populations that have a higher frequency of certain conditions. And we do have a screening available for certain populations, such as the Ashkenazi Jewish population, for a breast cancer risk. So just to, I guess, going a little bit more into detail about genetic testing itself. As I mentioned, it may or may not be prescribed in a genetic counseling session. It would really depend on if it would be informative and if there's informed consent and the family wants genetic testing. And a lot of times genetic counseling can take place after a genetic test has been conducted. So this could be ordered by the primary physician or a specialist, and then depending on the results and the interpretation, the genetic counseling session could take place afterwards. And what's important about the genetic counseling session following a genetic test is that the genetic counselor can help you understand what this genetic test means, what exactly was tested, if there's a way to perhaps understand how the disease will develop, and also help and provide support for how the decisions surrounding these results can be made. And, you know, I guess all of you can imagine sometimes having a result that's undesired could be very difficult to deal with. And we are also trained to provide support, emotional support for that. In neurology especially, we do a blood test, but in other areas of genetic counseling, we can do saliva samples. If it's during pregnancy, there could be a collection of the amniotic fluid as well for genetic testing. So what can you expect in a genetic counseling session? Perhaps some of you have already had one, but if you haven't, this is just to say that it lasts about 1 hour. Typically, pre-COVID times, they would be always in person, unless the person lived very remote. But now we've moved along to doing most sessions virtually or by phone. We prefer not to work by phone because you can't see the person and assess emotions. And also, we have a lot of visual aids. So doing it virtually is much more helpful, like as we're doing now. The session usually begins by taking a medical history of a personal medical history of the person who's seeking the genetic counseling. And then we'll go into a detailed family history to just get an idea of the amount of information we could collect and do a risk assessment. And of course, all of this is done with the utmost confidentiality. Once we've collected enough information, or the information that we need, we can help sometimes determine risk just by this. Even before doing genetic testing, we can give a risk estimate of a person either inheriting a genetic change or mutation, or developing the disease. And then we can also start to get an idea of what the inheritance pattern is. So how the diagnosis or whatever it is specifically that we're looking at is being passed down from generation to generation. And then with this information, we can see if genetic testing would be beneficial, and then move forward with that. And then if ever genetic testing is conducted, we have a follow-up session as well. And of course, all of this is done with informed consent. If an individual is unable to consent for themselves, we work with the person responsible for their medical decisions. Okay, so now I will switch gears to talk a little bit about genetics. I don't know if anyone has questions before I move on, a little break. It seems like everyone's on board.
And people feel free to write your questions in the chat if you can. Otherwise, we'll follow Nelly and Christiana's lead on when they wish to take questions.
So actually, this is my favorite part. I love genetics. So I guess like as a little crash course, many of you probably already know that we have many different cells in our bodies, and each of our cells have different roles. So we have cells in our eyes that are responsible for helping us detect light and see color. We have cells in our liver that break down different compounds for us to be able to have energy. We have cells in our brain, our neurons, that help us understand the world around us. And each of these cells get their instructions to carry out their functions with DNA. So we've all heard of DNA as well. There is a lot of DNA, so much so that if you take all of the DNA on our bodies, so in one human, and you stretch it out, it would go from here to the moon 365 times. So there's a lot of information there, and there's many genes. In fact, yeah, there's over 25,000 genes that we are estimating at this time, and we know the role of about a quarter of them. So there's a lot that we've learned a lot. We've made great strides in genetics in recent years, but there's still a lot to learn. And there's so much information in our DNA that it has to be tightly wound up into structures called chromosomes. Maybe you've heard of chromosomes before, so that each of our cells can actually pack all this information in. And humans have 23 pairs of chromosomes. So in terms of how DNA plays a role in different diseases, this pie chart could represent, most of the time, this could be applicable to many different diseases. It could be applicable to cancer, but of course, today we're here to talk about ALS. So most of the time, in the majority of cases, what we see is the sporadic form of ALS, which means that one individual in the family has a diagnosis of ALS, and it usually occurs after the age of 55. In contrast to where the genetic counseling really lies is the hereditary form of ALS. So this is the smallest percentage of ALS cases. And this is really to describe the situation where ALS in a family, and so there's more than one person that could be affected, is caused by a single change in a gene. And we can trace this gene through generation to generation. Now, if we have multiple members of a family that have a diagnosis of ALS or perhaps dementia, they could be related, and I'll explain why in a couple slides, then, and we cannot specifically pin down the gene that's involved, this would fall within familial ALS. So we know that there's something that may be genetic in this family, there may be a predisposition, but we cannot understand specifically the underlying cause at this time. And this could be due to either multiple genes acting or interacting with environmental factors. And it does increase risk, but it's more difficult to predict the pattern within a family. Oops. There we go. So for familial ALS, which is the category I was just describing, a good way to visualize this is to think of this jar here that represents a diagnosis. If you fill up the jar, you would get a diagnosis of a condition such as ALS. And then we have genetic factors that we're born with that we don't choose. And we have environmental factors that at this time are still unknown. So we can either be born with a certain amount of genetic factors, and then the environment can interact and fill up our jar that will lead to a diagnosis of ALS. Or we can be born with a higher predisposition for ALS, and then also be exposed to certain environmental factors that will bring us to develop the condition. At this time, the environmental factors for ALS remain unknown. Hopefully this will change with more research, of course. So if we do see that an individual with ALS has a family history, how can we start to distinguish between a familial form of ALS where we can't really know the cause and a hereditary cause for ALS? So even though, I forgot to mention here, but even though all these three categories are different in terms of the reason behind why ALS manifested, the way that they look in the clinic look pretty much the same. So it's difficult to distinguish based only on clinical symptoms. However, in some cases, when it is a hereditary cause of ALS, what we would expect to see is that more than one individual in the family will have a diagnosis of ALS or dementia, specifically frontotemporal dementia. And these relatives will be on the same side of the family, so they would be related. And what we would also expect to see is that the way that these individuals would present themselves in this family would be through vertical transmission, meaning it would pass from grandparent to child. And sometimes this is not always the case for various reasons, but sometimes there could be an earlier age of onset when it's hereditary. So before around 5 years before the average age that we would see in the population. So here's just an example or visual diagram explaining what I just said. So here we have a couple. So all of us, out of the 25,000 genes that I mentioned that are estimated that we all have, that we all share, we have 2 copies of each of these genes. One that we inherit from our mothers and one that we inherit from our fathers. And when we have children, we pass down one of these 2 copies to our children. So in this diagram, we have a dad here that has no ALS. So both of the copies of his genes that have been related to ALS have no changes or mutations. And then here we have mom who has a diagnosis of ALS, and we identified a change in one of the copies of her genes related to ALS. So when they have children, dad passes on one of his genes and mom will pass down one of her genes. And it could be either one, the one with the change here in blue or the one without the change. So if she has a daughter here that did not inherit the change in the gene, her risk for developing ALS is the same as the general population. We never say 0 because no one has 0 risk for anything. Life is risky within itself, but she will not be at increased risk. Her son, however, who inherited the copy of the gene that's related to ALS with a change or a mutation, will eventually develop symptoms. So all of this being said, we started off with really distinct categories, but in reality, theory and reality is not always the same, especially in medicine and science. We have been finding that there are 10 to 50% of sporadic cases of ALS, so that don't necessarily fit that description of having multiple family members that have ALS with the vertical transmission and earlier onset. We don't see this, but within these sporadic cases, 10 to 50% do have a hereditary cause. So a gene was identified in these individuals. So because of this, genetic testing, particularly at the neuro, has been pushed forth in a good way to test all individuals that have been diagnosed with ALS, because sometimes the ones that seem to be in a sporadic category are actually in the hereditary category. And this can happen because sometimes there's not enough opportunity for the disease to present itself. So either it's a small family, perhaps someone died accidentally or prematurely before they had the opportunity to develop the disease. And also sometimes, depending on the genetic variant, an individual won't fully show symptoms until maybe later in life, or maybe not show symptoms at all, although that is very rare. So as I mentioned, prior to, I guess, this presentation today, or maybe some time ago, genetic testing was not indicated for all patients with ALS without a family history. It was only indicated for patients with ALS who did have a family history. However, a new gene was discovered that changed this. And actually, we'll talk more about this in a couple of slides, but the C9orf72 gene is the most common hereditary cause for ALS. So once this was discovered, they realized that it would be important to test everyone, especially due to the fact that clinical trials are becoming available for certain individuals who are diagnosed with ALS and that are carrying certain genetic changes. So for what's important, I guess, to consider before genetic testing is first, there's two categories. I'll talk about diagnostic testing first and then predictive testing. So for diagnostic testing, this means that an individual has received a diagnosis from ALS by their neurologist, and now they want to see or rule out or see if there's a hereditary cause. So there is a possibility of identifying a genetic change. If you remember from the pie chart, the possibility of identifying a genetic change is the smallest possibility. Most of the time, we don't detect, we don't find any genetic mutations or the actual reason for the ALS. However, if we do detect a mutation, this has important implications for relatives and future generations, as you can imagine. And because clinical trials are becoming increasingly available, it does provide some hope for individuals living with ALS. And sometimes there's this conflicting emotion of wanting to wanting to find the cause of the ALS and seeing if there's a clinical trial available, but then also having to live with the idea that this might affect their loved ones. So we help sort of hold on or tease apart the duality of these complicated emotions. The possible outcomes of genetic testing. So usually there are three. Most of the time, the results are negative, which means that we did not detect any changes in the genes tested that have been previously associated with ALS. And this is really based on the current knowledge that we have now. So it is a rapidly expanding field, genetics, and new genes can be added every so often. So when we say negative, sometimes it is really for this time. It could change and it could lead to more testing in the future if new genes are discovered. A positive result in genetics means that we detected a genetic change in one of the genes that were tested. So this can help confirm a diagnosis, especially if the genetic change is related and has been reported with the same diagnosis. And this opens the door not only to clinical trials, but to testing other relatives in the family, even the ones that don't necessarily have symptoms. And then there's the third possibility, which I think all genetic counselors hate, including and patients, and it's when we have a result, but it's inconclusive. So we did detect a change, but we cannot interpret whether or not this change is causing the condition in question, or if it's just a natural change that sometimes happens in our genes. So this can leave us in a state of uncertainty. Sometimes genetic testing can bring more questions than answers. But usually what we do in these cases is ask the patient and their family to recontact us in 3 to 5 years, because hopefully there's some information that could come of this. And then eventually results that fall within the uncertain category will be segregated into either negative or positive. It won't stay uncertain forever. Now, the other type of genetic testing that we can offer is predictive testing. And this one is a little more complicated, it involves a little more steps, because it involves individuals who have a relative that has been diagnosed with ALS, but that don't have symptoms themselves and want to understand their own risk and maybe perhaps plan for the future. So the first thing first, what we do is assess whether or not a gene has been identified in that family. So if we have not determined that the ALS within this family is hereditary and the cause of the ALS has not been identified, we always go by testing the person with the diagnosis first. We don't start in a family to test individuals that don't have a diagnosis, because it would not be informative enough for us. So that's the first thing. If a genetic change or mutation has been identified in the family that explains the diagnosis of ALS, then we initiate what we call a predictive protocol. So we have several steps involved where we first have a phone call with this individual, then have a meeting, then see if the test is really something that would benefit this person's their well-being and their life. And then after we eventually give the results and have a follow-up session afterwards. And just some things to consider for the genetic testing is that first, questions around insurance have often come up during this time. So it's just to mention that sometimes we do encourage individuals to sort out their insurance before coming in to the genetic counseling and doing the test. But that being said, there are also laws protecting individuals from still accessing this test but not being discriminated against. So there's the Genetic Non-Discrimination Act, and this is just so you know for that your rights. That if someone asks for your genetic information, if it's an employer or insurance company, it's illegal and you're protected under this act. That's not information that you should be asked to be giving out. Other individuals who are interested in predictive testing is to plan for their life. So to decide whether or not to have children or how to assure that their children are properly cared for if ever they get sick. What to do with work. A lot of people, retire early and take that trip that they always wanted to take. And it is just really important to mention that at this time, predictive testing is really based on having that information for oneself. It doesn't impact at all medical decisions because there is no cure, and there's no prevention or any way to alter the course of the disease at this time. And hopefully this will change with research because there are early clinical trials that are developing now. And Christiana will tell you more about this. This is really much more her area. But there are early clinical trials where they're looking at individuals who are carrying genetic changes for ALS, but that have not yet developed symptoms. But again, this is very much in its infancy, but it's a start. So just to briefly discuss the genes that we usually test in ALS, because this whole time I've been talking about genes and there are changes in genes, but we haven't named them yet. So the most common genetic change associated with ALS is the C9orf72. So this is a gene that's important in the central nervous system, and it's been found to cause ALS in around 40% of individuals who have a family history of ALS, and almost 8% of individuals who have no family of ALS and who have been diagnosed with ALS. Then there's the SOD1 gene. So this one also works in the central nervous system, and it's quite common, less than the C9orf72 gene, but still quite common, seen in around 20% of hereditary cases and 3% of sporadic cases. And then there's another popular key player of the genes for ALS is the ataxin N2. So this is a gene that's a little bit different than the two that I just mentioned, not only because its function remains unknown, but because it's not a causal gene, which means that having a change in this gene does not necessarily mean that ALS will be manifested. It is a risk factor, so it could increase the risk of developing ALS, but it's not as black and white. And then there are also many other genes that have been associated with ALS, as well as dementia, that are sometimes tested for depending if there's a family history or not. So what I mean is that here's a decision tree on how the genetic testing at the neuro will be done. So if someone has a new diagnosis of ALS, first we distinguish between if they have relatives that have ALS or if it's just them and their family that have ALS. However, at the first step in either case, we test for C9orf72 because of how common it is. And if it's positive, it stops there. But if it's negative, meaning that there were no change and the C9 gene seems similar to individuals in the general population, then we move on to the next step. So if it's in individuals who have are the only ones in their family who have ALS, we test two more genes, the SOD1 and ataxin-2, and that's because there are clinical trials available. But if it's an individual who has other affected family members, then we'll go broader and test for more genes. And with that, I'll pass it on to Christiana, and I'll stop sharing my screen here.
Thank you, Nelly. There's a question in the chat that I don't know if you want to address it while I get my PowerPoint up.
Yes.
The question says, if someone has a close family member, a father, who died of ALS without being tested for genetic form, would predictive testing be useful? Thank you.
That's a very, very good question. So sometimes this does occur, unfortunately. So it depends if there's more than one person in the family. If it's the only person, then it would be difficult to do predictive testing, because having a negative result, it wouldn't necessarily be a true negative result. It could be falsely reassuring, is what I'm trying to say, because we first need to determine if the case of ALS was hereditary and what type of change was found in this individual before testing someone who doesn't have the condition. However, this is something that is up for debate as well, because sometimes if there are no other options, maybe getting that test would still be helpful. So I think it really depends on each family's situation, and then talking with the genetic counselor and seeing what type of information can be extracted, and if it would be actually informative and helpful.
Thank you.
Can you see my slides? They're up?
Yes, we see it perfectly. Thank you, Christiana.
Thanks so much, Nelly. And if there are more questions, that come to mind right away, feel free to put those in the chat, and then we'll get to those after these few slides. So for those who may not know me, my name is Christiana Salmon. I work at the Montreal Neurological Hospital, although I'm now based in Toronto as I'm working on a national project. You may know me from running clinical trials at the Neuro for many years, and I'm now working on a Canadian strategy to help improve access to genetic testing for patients living with ALS. As Nelly alluded to, our understanding of ALS genetics has changed a lot. There are many clinical trials available, and while we're quite fortunate in the province of Quebec to have easy access to genetic testing, or relatively easy access through your neurologist, that's not the case in many other provinces across the country, and so there's a big gap in equity that needs to be addressed in this area. So I'm briefly going to touch on some aspects of ALS genetics specifically. So Nelly alluded to these different buckets or categories of patients from a genetic standpoint. So she mentioned familial, hereditary, and sporadic. But if you've listened to webinars in the past or read about ALS online, you might have seen them lumped into just these two categories of sporadic and familial. And so when we talk about familial ALS, this little pie chart on the right, we're really referring to those patients who have a clear family history of the disease. Although today our understanding and our knowledge of what exactly is a family history is changing. So this term is becoming more and more arbitrary. But when we say familial ALS, we're referring to patients with a family history typically. And then when we say sporadic ALS, we are referring to patients who have no family history, so the case appears to be isolated, and that accounts for historically the large proportion of ALS patients. However, in the familial ALS patients, even though there is a family history, there are many families who have ALS that we can see a clear pattern of inheritance through generations that have no known cause. So we have not identified what the genetic change is yet that is causing ALS in those families. And that's the purple part of the pie chart on the right. And in the sporadic population, you'll also see that there is a proportion that have known genetic causes despite having no family history and appearing isolated. And so, as Nelly mentioned, familial and sporadic ALS are clinically indistinguishable. Patients will have a similar course of disease. Symptoms will be similar at onset. There will be bulbar onset patients and limb onset patients. And so really it's by genetic testing that we can make this distinction. And as I said, there are unknown causes in the familial ALS population, and some sporadic patients have a genetic cause. And why is this important to understand that even though we are making this distinction of familial and sporadic, in the underlying genetics of everyone, the more that we understand about the genes that we know cause ALS or are associated in increasing risk of ALS, the more we can understand about ALS as a disease as a whole, and that is really important. And so this is a really complicated slide only to show you that these are all of the genes in which we've discovered changes associated to either causing or increasing risk of ALS. But that's not just restricted to patients who have a clear family history of the disease. And when we look at the underlying mechanisms that occur in our body related to these different genes and what their natural function is in our body, they're all related. And so the more we can understand about genetics in ALS, the more we'll understand about ALS as a whole disease. And so this chart shows gene discovery in ALS over time. It is outdated by a few years, but I do like it because it also gives us more information. So you can see here that the amount of genetic mutations or genetic changes associated to ALS has really been rapidly evolving over time from the discovery of the very first change, which was in the SOD1 gene. And this is largely due to better studies, better genetic testing technology, the ability to do whole genome sequencing. And then the size of the circle is representative of how important that gene is in ALS. And so Nellie already mentioned that the C9orf72 repeat expansion is associated with, quite a large incidence in individuals who have no family history. And so that's a quite important mutation for us to pay attention to. And there are there are many of these mutations that have therapies that are being developed right now. And so for the SOD1 gene, the FUS or FUS gene, ataxin-2 repeat expansions, and C9orf72 repeat expansions as well. And when we put those into numbers, this is something I always like to show people. In the sporadic population, these may seem like very small numbers. If we say that 2 to 4% of sporadic ALS patients, so there's no family history, might have a mutation in the SOD1 gene. That's a small incidence. And if you look at FUS and ataxin-2, less than 1% as well. But when you start to combine all of these mutations together, there is the potential that 10 to 15% of patients with no family history, so there's no identified hereditary cause, no one else in the family has ever had ALS, could have one of these genetic mutations or changes for which therapies are currently being developed. And that's really important for us to recognize, because we'll only know this by doing genetic testing. And so I want to talk a bit about genetically targeted therapies, because we always get a lot of questions about this. I'm not going to speak about specific therapies themselves, although you can ask a question in the Q&A, most definitely, just because there are many companies using similar strategies for the same genes, which is very exciting. So I'm going to talk about how these types of therapies might work. And so genetically targeted therapies in ALS will target a specific genetic mutation. So just a recap of what Nellie has already mentioned. A genetic mutation or genetic alteration or change is like a mistake in our genetic code. And so sometimes it's a single change in the code. So let's say you're reading a sentence and one word is changed or one letter. That can have a large effect. And sometimes it's what we call repeat expansions, which is addition of genetic code that's not supposed to be there that can result in disease. And so genetically targeted therapies will try to target these changes, and will do so by a variety of strategies. And before I get into those, a quick reminder what Nellie already mentioned. Our DNA is our code of life. It codes for, the fact that I have brown hair and brown eyes, and someone else might have blonde hair and blue eyes. It accounts for the fact that I'm female and someone else is a male. And it also codes for all of these things called proteins. And that is the most important part, is our DNA codes for all of the proteins that do everything in our bodies. They make up our cells. They move things around the body. They have important functions in our everyday life. And these proteins we call them the building blocks of life. And this is an important concept to understand, that DNA ultimately codes for protein. And so the first type of therapy that can be used to target genetic diseases are called antisense oligonucleotides. That's a really big word. We'll call them ASOs for short. It's a little easier to say. And that big scary word just means that they're short synthetic pieces of DNA. So they're not true DNA that have come from a human or been extracted for a human. They're synthetic pieces of DNA that have been made in a laboratory. Because they're made in a lab, they're not stable over long periods of time. So that's why you would need repeated doses. And their aim is to either reduce, restore, or modify protein function. And I'll use an illustrative example in a moment. There are several examples of approved ASOs for other diseases. The most relevant one that you likely have heard of is possibly Spinraza for spinal muscular atrophy. Spinal muscular atrophy is a disease of mostly infants, although it can affect people later in life, where motor neurons, the gene coding for motor neuron survival is defective. So motor neurons die and these babies don't develop their muscles properly at a very young age. And so Spinraza is an approved antisense oligonucleotide, which restores that function of the defective DNA. Antisense oligonucleotides cannot cross the blood-brain barrier. So our brains are protected by something called the blood-brain barrier, which is a very dense layer of epithelial cells that prevent pathogens like viruses and bacteria, just things that aren't supposed to be there from getting in. And so because of that, ASOs are typically injected intrathecally by lumbar puncture. So patients have to have a spinal tap at repeated individuals because, as I mentioned, the synthetic pieces of DNA aren't stable over long periods of time in order to get the treatment. And so I'll use an illustrative example using the SOD1 gene. So in our body, the SOD1 gene codes for a normal functional protein that, let's say, looks like this. It doesn't actually look like this, but we'll say it looks like this. And so our DNA codes for this protein. That's what it's supposed to look like. It's this tightly wound little ball of amino acid chains, and it has a certain function in our body involved in antioxidant reactions. And this function is dependent on this shape. When we have a mutation in the SOD1 gene, so we have a single change in one part of the DNA sequence for the SOD1 gene, that one little change results in a protein which doesn't fold properly. So let's say it looks like this instead. So it's big, it's got some little appendages at the end, it's not tight and wound up like it's supposed to be. And because it forms this misfolded shape, they kind of clump together and stick to each other because they're not folded nicely and they hook onto each other, and they form what we call these aggregates in the body. And it's these accumulation of these aggregates of misfolded SOD1 protein that evidence tells us causes ALS in individuals who have mutated SOD1. And so what an antisense oligonucleotide treatment will do, it will come and bind this little synthetic piece of DNA, bind to the strand of DNA where there is that mistake. And so if we're blocking that mistake, then we're just going to block protein from being formed entirely. And then we won't get these clumps, and ideally we won't get disease. And so there is a very late stage clinical trial in phase 3 for this kind of strategy right now, and we're pending results this fall, which is very exciting. However, as I said, this protein has a normal function in our body and it's supposed to be doing something. And if we're just blocking it out with an antisense oligonucleotide, we're blocking the good protein that can be produced as well. And so that might be an issue, which leads me into the next type of therapeutic strategy. So antibody therapy is another type of therapeutic strategy that can be used. Antibodies are proteins that are in our body that are used by the immune system to identify and neutralize foreign bodies. So if you get an infection, like a bacteria, your body recognizes that that bacteria is not supposed to be there. And these antibodies, which are shaped like little Ys, will come in, bind to the bacteria. So they'll stick to the bacteria and the body will say, oh, hey, that bacteria is not supposed to be here, and get rid of it. And that's how we fight infection. Antibodies can be synthetically produced in a lab. So therapeutically, they could be developed and made to target specific things in our body, and then kind of hijack our natural immune system to combat disease. And so we're going to use our same mutated SOD1 example to illustrate this again. We have this mistake in the genetic code, which is causing misfolded SOD1 protein, which is the problem because it forms these clumps, sticks together, and causes ALS. If we can develop an antibody which binds to the misfolded SOD1 specifically, it can then flag to the body that this misfolded protein should be eliminated by using our natural immune system. So there's a potential advantage in using antibody therapies over antisense oligonucleotides. One, because they can be injected intravenously, so you wouldn't need a spinal tap because they will cross the blood-brain barrier. And two, this antibody would be specific for the misfolded protein and not the healthy, correctly folded protein, which is also in our body. So it would be targeting just the misfolded protein. The challenge with antibody therapies is that it's very challenging to develop these therapies to be the antibodies to be extremely specific in this manner. But there is an ongoing clinical trial using this strategy as well. The final therapeutic strategy that can be used in ALS is gene therapy. There is one other, but it's related to these ones, so I won't discuss it today as it's rather complicated. But gene therapy is probably something that many of you have heard about as somewhat of a hot topic, especially in neurological diseases, where it replaces or corrects a faulty gene entirely. So gene therapy will use a viral vector to introduce the corrected gene. What that means is that they'll take viruses that don't cause disease in humans. Usually we use adenovirus or adeno-associated viruses, AAV. They don't cause any disease in human. They're benign to us. But if you inject them into a human, they'll replicate themselves. And so you can insert a healthy gene into this viral vector to get it into humans, and then they'll make more copies of it. This is a very novel type of technology, and only a handful of approved therapies in gene therapy exist, all of them in pediatrics, which poses a problem for adult diseases. The most prominent example is probably Zolgensma, again for spinal muscular atrophy. So this is a single infusion that's given. The viral vector introduces the correct gene because there's a faulty gene in spinal muscular atrophy. And we're a few years post-clinical trial now, in these children seem to be developing normally and meeting all of their motor milestones, which is very exciting. And this is more challenging in adult diseases, however, because our immune system is much more developed and advanced than in pediatric diseases. But there are several companies working on gene therapies for ALS right now. And so using our mutated SOD1, again, as an example, we have our DNA with our little mistake in it, which is causing catastrophic reactions of these misfolded proteins. When we use gene therapy, it would be an intravenous infusion, which would introduce the viral vector that contains the healthy gene. The healthy gene would then be produced. And so we then have healthy DNA with no mistake in our body. And so the correct protein can be formed. So just to summarize, because that's a lot of very complex concepts all at once, there are three main strategies for genetically targeted therapies for ALS right now. Antisense oligonucleotides aim to interrupt the mutated gene. Antibodies will flag the end product, which is the protein, for destruction by the body's natural immune system. And gene therapy, which is still very experimental, aims to correct the gene itself. And so drug development of genetically targeted therapies for ALS is really booming at the moment. And so this is just a snapshot of the companies who have a publicly declared pipeline for therapy specifically for the genetic forms of ALS. And so you can see, I'm not going to go through all of these individually, but you can see that there's a variety of the mutations that I've spoken about and a variety of the strategies. So antisense oligonucleotides with the Biogen programs, gene therapy, antibody therapy, and some recent players to the game who have not disclosed what their therapy is yet. And while it can be disheartening because the proportion of patients with a genetic mutation that we can target is small for ALS, this is also really important for us to better understand ALS, the disease spectrum as a whole, and how we can actually treat it. Because if we can start chipping away, piece by piece, our understanding of these genetic forms and developing treatments which can treat these forms, then we can apply those learnings and those concepts to all ALS patients and hopefully develop better therapies and better trials in the future for the broader spectrum of the disease. And so with that, we'll take questions.
Well, thank you very much. We do already have a question from Norm. How much has genetic testing improved over the last 6 years? I was tested back in 2015. Should I be retested in 2021?
It's a great question, Norm. Nellie, do you want to take that or do you want me to take it?
Go for it.
Okay, so that's a great question, Norm. So our genetic testing technology itself hasn't necessarily improved in that time frame. It's improved greatly over the years, but since 2015 and today, the technology is still very much the same. What could have changed is the genes for which there are clinical trials available that you may not have been tested for previously. And so if you were tested in 2015, there are definitely clinical trials for they're not new genes as if we just recently discovered them, but they're new clinical trials for genes that weren't in clinical trial when you got tested in 2015. And so it would be worth it to speak to your neurologist about potentially getting testing for these genes at this point.
Wonderful. Thank you very much. It is time for questions, people. You may either unmute your microphone and perhaps your video as well, or use the chat box to write your question. And so I'm going to give you guys a few minutes or a few seconds, actually. Minutes would seem interminable if we were to be quiet, for you to, or just raise your hand, use the reaction button to raise your hand, and then we would see that you wanted a question. I'm assuming
any questions are helpful because the webinar will be archived, and so patients who aren't able to attend and watch the webinar online might have the same question. So all questions are welcome. For sure. But if there are not going to be questions, we will not be staying on for ever and ever. So. Does anybody have a comment that they'd like to share with Christiana and Nellie?
Hi, it's Joanna, one of the community leads at ALS Canada. I have a client newly diagnosed. She lost her son to ALS last year, and he passed away very fast, within a few months. And now she was diagnosed with ALS. The family is terrified. There's one more brother. And I think there's some genetic testing happening at the Sunnybrook Clinic. I have to do a visit with them on Thursday. I'm sure they're going to have tons of questions. And one of the questions that they had for me on the phone was, what genes are we tested for? So I direct them back to the Sunnybrook Clinic, but they're not sure if they do the whole, they test for all the genes. But I guess my question is, how can I support them, right? From the community lead point of view, because I don't have a lot of medical expertise, and I'm sure directing them to the clinic is important, but they're expecting me to provide some sort of support as well.
Yeah. Nellie, do you want to start off with some comments from the counseling perspective?
Yeah, sure. So it sounds like you're doing important work. I think, well, have they had a genetic counseling session? Because there could be that their genetic testing was ordered by their neurologist, but they're seeking more information in terms of incorporating the results.
I don't think so. So probably that's the first suggestion I should have for them, right?
Yeah, that's really where, so the need that you just identified is exactly where the genetic counselor could come in. And then also just verifying if there is a family history. So making sure that everything was tested for and seeing what type of results they are. And even if they're negative, to be able to explain, that a hereditary cause was not identified, but perhaps there are some of the changes that would be revisited later. So it would be good to see the report and then also refer them to genetic counseling services. That's what I would say.
Yeah, and if I can add to that, so the son was affected by ALS first. There was no family history, and now the mother is affected, if I'm understanding correctly. It's rare, but it does happen. And I do recall a family that we have in Quebec where, you know, there was like 6 children in the family, all in their 40s, and one by one they started getting ALS, and there was no family history of the disease. And then the mother in her 70s finally developed symptoms. And so that was a big shock to that family. And I think, you know, when it's passed down from generation to generation, obviously emotionally that's a lot to handle as well, but those families like know that it's in the family, and there's some level of understanding already, which can help in the conversations. For something like this, that can be a big shock and very scary to them. But it's rare, but it can happen, because one of the issues with mutations in ALS is that there's this concept of penetrance, which I'm not going to do justice explaining, but it's kind of your likelihood that you'll develop disease if you have the mutation. And so in some diseases like Huntington's, if you have the mutation, you will develop symptoms. But in ALS, sometimes you can have the mutation, and as Nelly said, it's rare, but you can possibly never get symptoms. And that's what makes one of the aspects of genetics in ALS so challenging, and why we have individuals who have a change in their genetic code but don't have a family history. And so the other thing that I will say, you know, if they are ordering genetic testing, that's fantastic. If the son ever had, before he passed away, DNA banked anywhere through a research project, if he didn't see a genetic counselor, then that might be the only option. If the clinic or a research project banked his DNA, having DNA from the son to test as well could be really helpful, especially if they get an inconclusive result, or if they're not ordering a broad panel, it can help direct the genetic testing. And I know we're based in Quebec, but I can speak a bit to Ontario. I think the only neurogenetic counselors in the province are located at North York General Hospital. So if they are referred for genetic counseling, her name is Kristen Miller. She's wonderful. That's where they're located. And obviously it's up to the neurologist what they're ordering for genetic testing. I won't lie, it's challenging in Ontario. We're actively working on it with Dr. Shoesmith, Dr. Chum, Dr. Eisenberg to try to advocate with the MOH to get better access to testing.
Thank you. I know in the past, I have one more question. I know in the past, my families who had genetic ALS, genetic component in their ALS, they were tested in Miami. Are they still doing that?
So there is a research project ongoing in Miami called the PREFAL study. So it's pre-familial ALS, and they aim to follow individuals before they develop ALS, but who have genetic changes. So it's possible that they referred for that study, and then the testing was done through that study. However, it's a research project, and so there's an important distinction between research and clinical genetic testing. They're two distinct things, and research testing should not be used for clinical purposes. And so any research testing should always be reconfirmed with clinical testing from a clinical lab. So either a big commercial lab or at like a molecular genetics lab at a hospital. Nelly, did I miss anything there that you wanted to comment on?
No,
research versus clinical?
That was perfect. It's more if it's a positive result. If it's negative, usually the negatives is okay. But if there is a positive result, which could really alter someone's life, we usually want to check that. Even if it's from direct to consumer testing, sometimes you know, we know all these popular tests now, there could be a positive result that comes up, and we always check that with the clinical lab because there are certain steps that have to be taken, and it does take longer, and it's much more thorough for medical decisions.
Yeah, and I will add to for Joanna, so in Ontario, one of the big issues is if a sample has to leave the province, that needs approval by MOH, the Ministry of Health. And the problem is there aren't any labs in Ontario who do genetic testing for ALS in the province, except North York General, which has a C9 or F72 repeat expansion test, but they only got their approval for that in December of last year. So it's relatively new. So what happens is you have to get this MOH approval. If there's a family history, it should be approved. However, it can take 3-4 months, and patients with ALS don't have that time. And if there's no family history, or the family history is unclear, we do have some cases that the physicians have brought forward where the MOH has denied the approval for testing because they've said there's no family history, you don't need to do that testing. So that's the mindset that we're trying to change right now.
Thank you. This is so informative. Thank you so much.
You're welcome.
Do we have any other questions? We have a comment. Great presentation, very clear and informative. Thank you very much. Thank you, Maris. That's nice to hear. Do we have any other questions or comments for Christiana and Nelly?
I guess maybe I can mention here that we do have a short feedback form, just so that we can improve every time and see if we hit all our notes and questions that maybe some of you may have. So I'll just, can I copy paste it in the chat?
For sure. And if you send me an email, what I can do is also send that link to the participants in an email after this. It will probably get done tomorrow morning because I have a meeting right after this conference. That will take most of my absolute pleasure.
Perfect. And we've tried to include content that won't get outdated really quickly, because this is a very rapidly evolving field. So if there's something that you feel that we should have covered that we didn't, please do let us know, because that's also very helpful to us.
Thank you very much. Well, I think it's been an extraordinarily informative and interesting conference. I've learned a lot just about the ideas of genetics. Thank you very much, Nelly, for that, and for what's related to research at this moment, Christiana. So I'd like to thank you both for your participation. This conference will be held again on October 12th in French at the same time from 10:30 till noon. So you will be able to repeat this. And I thank everybody for their participation, and we shall see you next time. Have a lovely afternoon, everybody.